reflecting our shared commitment of learning, growth, and the exchange of ideas. On behalf of Faculty of Medicine Universitas Indonesia, I would like to extend our warmest welcome and sincere appreciation to our speakers, who has generally taken time to share their knowledge and experience with us today. Lectures like this provide a valuable opportunity to connect academic learning with real-world insight, broadening our perspective beyond classrooms. Allow me to briefly outline today's program. We will start with the lecture sessions, followed by a question and answer segment, where you can all actively participate, which is highly encouraged, and engage with our speakers. Now to all the audience on site, please all be standing up for Indonesian National Anthem. Please be seated. Ladies and gentlemen, before we proceed to the main lecture, it is my honor to invite a distinguished leader of our academic community to deliver an opening address. Please join me in welcoming the Research Manager of FMUI, Dr. Budiman Bella, Specialist Microbiology Clinic Consultant. To Dr. Budiman, the floor is yours.
Distinguished speakers, Prof. Peter Lobie, Prof. Dr. Hariyono, and according to the list that I got here, Professor Ina is actually supposed to be with us. Therefore, I would like to welcome all of you to FKUI to become our speaker and in this event, which is very distinguished. We see here that the title is Advances in Molecular Oncology and Hormone-Driven Cancers, Implications for Biomedical and Clinical Research. As we know that this is a topic that is very important for Indonesia, especially in Jakarta. We know that cancer is indeed increasing in our country, and of course we would like to know what is actually behind these increasing numbers in Indonesia. And for the Faculty of Medicine, this is truly an honor for us to have our distinguished guest speaker, Prof. Peter Lobie. Thank you for being available to become speaker in this event, and we welcome you at Imary building, which is a research center that has been developed and constructed for the advancement of science and education at the Faculty of Medicine, University of Indonesia. So I believe that all of us will be obtaining a significant and substantial knowledge from this event. And of course, I really appreciate that there is a big interest from all of our students and our researchers to attend this very, I suppose, like significant meeting, that we will get information that will be very beneficial for all of us. So without further ado, I would like to welcome all of you, and I hope that we can enjoy the meeting. And of course to Prof. Lobie, we hope that you will enjoy your stay here in Indonesia, and we believe that we will extend collaboration in research and education between our institutions. So if I'm not mistaken, this is from Tsinghua University. Yes, we have quite a few, well, I can say not just a few, but a lot of collaboration with Tsinghua University. So this will be increasing our collaboration, and hopefully we will look forward to more fruitful collaboration between Faculty of Medicine, University of Indonesia with Tsinghua University. So without further ado, I would like to welcome all of you, and congratulations for this event to the organizing committee as well. Thank you, and I would like to hand it over again to the Master of Ceremony.
Thank you, Dr. Budiman, for insightful remarks. We truly appreciate your words of encouragement and support. Before we move on to the next session, it is my pleasure to invite the head of Master's Program in Biomedical Sciences, MPBS, to deliver a few welcoming remarks. Please join me in warmly welcoming Professor Dr. Wawaimuli Arozal, MUmed, PhD.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome to the International Webinar Advance in Molecular Oncology and Hormone-Driven Cancer, Implication for the Biomedical and Clinical Research, organized by Master Program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia. On behalf of the entire MPBS team, and as the head of MPBS program, it is my honor to welcome everyone here today. I would like to begin by acknowledge our distinguished Representative Dean, Dr. Budiman Bella, as a Research Manager of Faculty of Medicine, Universitas Indonesia. Thank you very much for your presence and your continued support for our program. I would also like to warmly welcome our three keynote speakers, Professor Peter A. Lobie from Tsinghua University, Professor Dr. Septelia Inawati from Biochemical Department, Faculty of Medicine, University of Indonesia, and then Prof. Hariyono Winarto from Oncology Department, Faculty of Medicine, University of Indonesia. And of course, I also warm welcome to our faculty members, and then students and also guests, wherever you join here by in person or virtually. Specifically, I would like to say hello to the students from the Sulan International Medical Faculty, Zhejiang Suren University, that already joined by online. So I would like to thanks to Dr. Wu Wen. Dr. Wu Wen is he's a student from a PhD student. He already invite the undergraduate student from Sulan University. Okay. Cancer remains one of the leading causes of mortality and morbidity of worldwide. Based on Indonesian Health Survey in 2023, cancer contributed to 0.6% disability in Indonesia. Biomedical science has a critical role to play in responding to the burden, not only in expanding our scientific understanding and advanced molecular research, but more importantly, in translating that knowledge into the better outcomes for the patients. Hormone-driven cancer, particularly in female reproductive cancer, represent a significant portion of the challenge, particularly here in Indonesia. And yet, our understanding of the molecular mechanism driving this cancer continues to evolve rapidly. We are deeply fortunate to have three distinguished speakers today to share their time, knowledge, and insight with us. Prof. Peter Lobie will open our session with a discussion of novel therapeutic in female reproductive cancer, bringing a globally recognized perspective on how emerging treatment approach and reshaping what is possible for the patient. And then Prof. Ina will then take us into the cutting edge of precision medicine, specifically how CRISPR-9 gene editing technology is advanced breast cancer therapy. And then last but not least, Professor Hariyono Winarto will discuss the clinical situation and clinical epidemiology of ovarian cancer. This event would not have been possible without the support and from the International Visiting Professor and Guest Lecture Program through the UI Insight Grant. So this grant reflects a broader institutional commitment to international collaboration and advance in biomedical sciences. So actually this program consists of 8-day activity. So from tomorrow, we will have a community engagement with the student in high school student, and then we also have some discussion with the industry, pharmaceutical industry for tomorrow. And then on Friday, we will have another guest lecture in RIK Depok. So we will invite our undergraduate student, and then there are also another one lecture from our alumni of PMIB, also Dr. Hadi Mulya. He's from Calgary University. And then, and then we also will have a visiting to the industry company, Etana company, on Friday. And then on Monday and Tuesday next week, for 2 days, we have a kind of coursework, hands-on work with the SGTA cluster in IMERI. So we will have a kind of stem cell introduction. And then we also have that today marks the beginning of lasting connection and collaboration between our institution and the global research community. It is my pleasure to officially open this international webinar. Let's have a wonderful and productive morning. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Prof. Muli, for such an inspiring, it's a really packed agenda for the next 8 days, apparently. We truly appreciate the valuable perspectives and guidance shared with us today. May the ideas conveyed encourage fruitful discussions and enrich our knowledge throughout the sessions ahead. We are now about to begin the lecture. The session will be moderated by Dr. Ria Margiana, Biomed Specialist Andrologi. Allow me to briefly introduce our moderator. Dr. Ria, now serving as the head of anatomy concentration in MPBS FMUI, had her bachelor, master's, and doctoral degree from Faculty of Medicine, Universitas Indonesia. She is also coordinating many research in so many fields of significant remarks. For example, impact of paternal age on DNA fragmentation index and its consequences for embryo development following intracytoplasmic sperm injection. Without further ado, please welcome Dr. Ria to moderate the session.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, distinguished professors, respected speakers, college lecturers, and also students and all participants, whether online or on-site. On behalf of the Master's Program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia, it is my great pleasure to welcome you all to today's international webinars. Thank you very much for joining us today. We are truly delighted to have participants from various backgrounds and institutions all coming together with the spirit of learning, discussion, and of course for the global collaboration in biomedical sciences. Okay, so let's just start it by introduction first for the first speaker. So it is now our pleasure to introduce our first speaker, Prof. Peter E. Lobie. Can you please the slides? Yeah. Prof. Lobie is currently affiliated with the Shenzhen International Graduate School, Tsinghua University. He earned his PhD in molecular endocrinology from Karolinska Institute in Sweden, and previously completed his medical and medical science degrees with distinction and first-class honors at the University of Queensland, Australia. Throughout his career, Prof. Lobie has held several prestigious positions, including Chair of Breast Cancer, funded by the Breast Cancer Research Trust, and as the co-director of the Center of Precision Medicine and Healthcare Research, and a professor and senior principal investigator at Cancer Science Institute of Singapore, National University of Singapore. He is widely recognized as his important contribution to cancer biology, molecular endocrinology, and precision medicine with an age index of 62. Wow. So his research includes work on pancreatic ductal adenocarcinoma, triple negative breast cancer, and molecular mechanism regulating breast cancer progression. We are truly honored to have him with us today. So ladies and gentlemen, please join me in welcoming Prof. Peter E. Lobie. Professor, the floor is yours for about 20 minutes. Thank you.
Thank you. So thank you for the kind introduction. Could I get my slides up, please? So I'm very happy to be here today. I'd like to thank the Faculty of Indonesia for their kind invitation. A special thanks to Professor Muli for her organization to bring me here. She's been really quite amazing and efficient in getting things done, so I appreciate very much. So as you've heard, I'm at the Tsinghua Shenzhen International Graduate School. It's a branch campus of Tsinghua in Shenzhen, more or less to promote the internationalization of science in China, so we're very open to collaboration across the world. So today I'm going to talk about one of the programs we have running in our laboratory, and we have quite a number of them actually, but I'll just talk about one that focuses on gynecologic oncology, which is a large problem in Indonesia. If you look at Indonesian cancer rates for females, the first three top cancers are breast, cervical, and ovarian cancer, which is quite different to the situation, for example, in China, where lung cancer is actually the primary cancer in women. Next slide, please. So I think I've been introduced. I graduated from Queensland in medicine, PhD from Karolinska, Spent most of my life at the National University in Singapore, so I'm very happy to be back in Southeast Asia. It's a very, very familiar feeling for me. I'm also sitting on the Academic Advisory Council of Tsinghua in Shenzhen. I have a position in another institution there. I've been elected a Fellow of the Royal Society of New Zealand, which in China is a very high status symbol, and also received the Chinese Government Friendship Award, which is the highest award given to foreigners by the Chinese government. So I've done what most scientists have done during their career, published a few papers. My hair color has changed and my H factor has also gone up. But also I have a large industry experience because we do a lot of translational work, especially related to drug development, and recently I've been appointed as an independent non-executive director of a Hong Kong stock exchange listed company. Next slide. Oh, sorry. This is my WeChat code if anyone wants to add me, and this is my email if you want to contact me. Next slide, please. So our lab focuses on understanding cancer biology across the whole spectrum, from development of the cancer all the way through to how cancer metastasizes and then how cancer recurs. And of course, once tumor recurrence occurs, the patients face a very poor survival outcome. Next, please. And so this recurrence usually occurs due to chemoresistance. The cells adapt to be able to survive in the presence of these cytotoxic drugs. They survive because of tumor heterogeneity. There are different clonal subpopulations within the tumor. And also both preclinical and clinical models are really not so good at predicting patient outcomes as of yet. And it's one reason why one of my students, I was just talking to Dr. Wu in Hangzhou, has set up a company to help predict patient responses to various drugs using organoid technology. Next slide, please. So to do these, or to achieve these objectives, the lab takes a pretty comprehensive approach. We have a lot of expertise in terms of mechanism. We're particularly interested in pro-survival pathways because if we can kill the cancer cell, it can't do anything else, right? We're not interested in static drugs. We're interested in drugs that can kill. We identify novel targets, identify regions of these targets which are involved, and then design first-in-class molecules. So this is a very dynamic field at the moment, as you might know. We've moved from cheminformatics to artificial intelligence design very, very quickly. It's really difficult to keep up. Every week almost it's changing at this point in time. And then once these drugs are developed, we translate them using various preclinical models before we go on to commercialize. And it's a necessary step, the commercialization, so that the patients can receive some benefit. Next, please. Just to say that the techniques in the lab are constantly evolving. Science is moving very, very rapidly at this time. Next, please. Okay, I am conflicted, just to put it up to cover myself legally. Next, please. Okay, so what I'm going to talk about today is one particular target called BAD, and we're targeting BAD phosphorylation, not as a kinase inhibitor like a lot of drugs do, but a drug that will specifically inhibit phosphorylation of BAD without impacting on kinase activity. Next slide. So next, and next, and next. Okay, thank you. So BAD is a bifunctional molecule. It stands for BCL2 associated death promoter. It's a member of the BCL2 family of proteins, very heavily involved in cell survival. So BAD is bifunctional. when it's phosphorylated, it acts as a promoter of cell survival by binding to a protein called 1433 and sequestering BAD such that BCL2, BCLXL, and BCLW can all bind to the pro-apoptotic members of the BCL2 family, promoting cell survival. When BAD is dephosphorylated, and we need dephosphorylation of only two residues, serine 75 and serine 99, dissociates from 1433, binds to BCL2, and releases the pro-apoptotic proteins to promote cell death. Next slide, please. BAD, of course, sits down of a range of, or a number of very important pathways in cancer, the RAS-RAF MAP kinase pathway and the PI3 AKT pathway, to result in phosphorylation of these two residues. The MAP kinase pathway, mainly serine 75, the PI3 kinase pathway, mainly serine 99. So there's already a large number of drugs targeting these pathways, but the issue with these drugs is while they're initially successful, they later go on to develop resistance. And if we take the example of vemurafenib, next please, we can see here in a patient treated with vemurafenib, a BRAF inhibitor, malignant melanoma patient treated, the response is really quite stunning. Next please. But within a period of 6 to 18 months, the tumour recurs. It comes back much more aggressive than before. Next, please. And the patient ultimately dies. And this recurrence or resistance has been shown to be mediated by BAD. Next slide. And again. Next. So we went ahead while we were still finalizing our move to Shenzhen in Singapore with two guys. Dr. Pasappa is now back in India, Vijay is with me in Tsinghua, and we designed a molecule which specifically inhibits the phosphorylation of BAD on serine 99. This causes loss of cell viability by enhancing apoptosis in cancer cells across a wide range of different tumours, but not in normal cells, and there is some reasoning for this, but I won't have time to go into it today. Next slide, please. So where can we use this drug was our next question, and one obvious one is in recurrent epithelial ovarian carcinoma, a very nasty disease. Its treatment follows standard cancer treatment protocols. There are a number of targeted therapies available, one of which is PARP inhibitors, which was approved by the FDA in 2017. But eventually, some cells do survive, they reproliferate back, and most patients experience a relapse with a bad outcome. So we thought this drug might be quite useful in this situation because ovarian cancer exhibits a very high level of phosphorylation of BAD, and also when the cancers become platinum drug resistant, the BAD phosphorylation levels also increase quite dramatically. Next slide, please. So a very good clinical fellow in my lab looked at this issue in combination with PARP inhibitors, using a series of patient-derived organoid and patient-derived xenograft models. So this is the patient. It was an end-stage, multiple chemo-resistant, multiple therapies. Nothing was effective. We took cells from ascitic fluid and cultured them for PDX. So N is our drug in blue, O is olaparib, the PARP inhibitor. We can see that they're somewhat effective as a single agent, but in combination we're able to obtain a very nice response whereby at least 1/3 of the experimental animals appeared to be cured So there looked like there was a pathological complete response. The tumour didn't recur. This made us very happy, but also at the same time, 1/3 is also not good enough. So I'll come back to this question a little bit later. Next slide, please. So she also looked in what is known as P10 deficient endometrial carcinoma. Most patients with endometrial carcinoma either have a mutation, a deletion or decreased expression by methylation in endometrial carcinoma. And when you lose P10, PI3 kinase pathway is increased in activation, so the serine 99 phosphorylation on bad is dramatically increased, in addition to PARP inhibitors increasing PI3 kinase activity anyway. So what she noticed was, and this was the same in ovarian carcinoma, that if she used this drug, this bad inhibitor MPB, we could decrease serine phosphorylation on bad, which then drove the apoptotic pathway towards cell death, but also at the same time MPB could prevent the increase in homologous recombination which occurs after PARP therapy. So PARP induces DNA damage in a synthetic lethal mechanism, but at the same time it also stimulates repair. So inhibiting bad phosphorylation was also able to inhibit the DNA repair, and it critically drove the cells into excess DNA damage and cell death. And we can see here in terms of this model of endometrial carcinoma that we were able to produce 100% complete response, which was for us also very exciting. Next slide, please. In the third type of gynecological cancer, we've also looked. We collaborated with a gynecologist from the Hong Kong University Hospital. There's a branch in Shenzhen, and we derived patient-derived organoids. And again, I'm sorry, the text is a little small, but we again showed that we could markedly improve the response of cervical cancer organoids to mitomycin C, which is an agent producing DNA damage and mimicking radiation damage, which is a common treatment modality in this type of cancer. And so using this experimental drug, NPB, I think we can see that inhibiting bad phosphorylation in gynecologic cancer might actually be very, very useful. Next slide, please. However, MPB wasn't so potent. It wasn't orally available. So we went back, and Esther spent quite a number of years developing a new analog of MPB called NCK, which was more potent, orally available, and continued to bind to bad, as shown here in this protease protection assay. But in addition, it also bound to 1433, which was maybe the reason for its increased potency. Next slide, please. She then went ahead with this new drug, NCK, and used it in a high-throughput screening strategy to identify potential drugs that may be useful in triple-negative breast cancer. I think we're all aware that combinatorial strategies in cancer is far more effective in treating tumor heterogeneity than a single agent. Next, please. So this is what the output looks like. It's an enormous amount of work to do one of these screens. It was very pleasing to see that she actually pulled out PARP inhibitors, which supported our previous mechanism. PI3 kinase pathways inhibitors, again, supporting our previous mechanism. But also on this side, the most synergistic compounds were these kinase inhibitors. both to c-KIT and c-MET. Next slide, please. So she took these two inhibitors. So one of them was crizotinib, which is an FDA-approved drug for ALK inhibition in lung adenocarcinoma, but it also inhibits MET, and OSI 930, which is a KIT inhibitor, a stem cell kinase involved in various cancers as well. It's stage 2, I think, at this moment, clinical trial, phase 2. So our bad phosphorylation inhibitor was able to produce a complete response in 2 out of 6 cases of triple-negative breast cancer. We were very, very happy with this, of course. Crizotinib improved it slightly, but combined with OSI 930, we were able to obtain a complete response in 5 out of 6 animals examined. Again, potentially offering a cure for a female reproductive cancer that is incredibly difficult to treat. Next slide, please. So now we can go back to ovarian cancer and use the updated version of our BAD inhibitor. And a PhD student in the lab, Vipul's an example of international PhD students that come to Tsinghua, Shenzhen. He came from India. We do welcome Indonesian students very much so coming to Tsinghua. No, even quite separate to any potential double degree master program. And through various omics techniques, he identified that a protein called PIM1, which is a kinase, was very, very heavily involved in the resistance or in the residual tumor, which persisted after bad phosphorylation inhibition and PARP treatment, PARP inhibitor treatment. PIM has a well-known role in cancer. It promotes a lot of the oncogenic drivers of cancer progression, but it's also known as a rescue kinase. So when we inhibit PI3K or ERK or MAP kinase pathway, PIM kinase steps in and says, I'll do the job instead. And so we saw not only slightly increased expression, but also increased phosphorylation. Next slide, please. And so I've left one of the best slides till last. So really, as I said, PIM is a rescue kinase. It uses an alternate pathway to mediate phosphorylation of not only serine 75, but also serine 99. But PIM kinase inhibitors are in clinical trial. They tend to be rather toxic. So we changed our protocol, only 3 cycles of therapy, 4 days each. But I think here in green, even you can see after the first cycle, the tumor basically disappeared and didn't come back. We were able to achieve 100% complete response using a PDX, patient-derived xenograft, for ovarian carcinoma, simply by 3 rounds of 4-day treatment, which was for us a really astonishing result. So I think, next slide, please. So I think you can see in the short time I've had to present that science can be very useful to identify new mechanisms and new treatment modalities. And so to summarize, simply inhibiting the phosphorylation of one serine residue in a protein with these small molecules was really sufficient to drive a complete response in a number of different female reproductive tumors in combination, which is the gold standard in oncology anyway. And so we believe, and we're going forward to commercialize this program in China. We actually have got funding from Hong Kong just last week to do it. and may be useful in a range of female reproductive cancers which are of particular relevance to Indonesia. Next slide, please. So I have a lot of people and a lot of collaborators to thank. It's a very large program that we're running. I'd like to thank Vijay and Jiangxi, my two co-PIs in the lab in Tsinghua, Shenzhen, as well as all the lab members and collaborators, not only from around China, but around the world. And hopefully soon we can add UI to the list here. So thank you very much. Again, I'm very happy to be in Indonesia, be back in Southeast Asia, and I'm happy to answer any questions, which I guess will be later in the discussion session. Thank you.
Okay, thank you for your amazing insight for us. It's so inspiring. We know actually there are so many mechanisms underlying how to help to give a solution to cancer problem. It's a very widely big case, not only in the world but also in Indonesia, of course. So let's move to the second presenter, which is Prof. Septelia Inawati Wanandi. Kindly introduce her, even though actually maybe 100% all of us know her, already know her. Okay, so actually Prof. Septelia Wanandi completed her medical degree at FMUI, obtained her Dr.rer.physiol degree from Philipps University at Marburg, Germany, and continue the postdoctoral, right? Yeah, postdoctoral training at Hokkaido University, Japan, and she also has international research experience as the visiting professor in Germany, including at the European Molecular Biology Laboratory in Heidelberg. She currently serves as the head of Molecular Biology and Proteomic Core Facility at IMERI FMUI, and is actively involved in several national and institutional committees, including Medical Research Ethics Committee at Dharma East National Cancer Hospital, the National Committee of Stem Cell Service, and National Committee of Material Transfer Agreement at Ministry of Health and of the Republic of Indonesia. She also served as Director of Medical Science and Technology at Mandaya Hospital Group and as the editor of Discover Oncology published by Springer Nature. What a good role. You can take this role from Prof. Ina. Her work has made important contribution in cancer genetics, biomolecular research, and personalized medicine, particularly in breast cancer-related studies. It is a privilege to have her here with us today. Please warmly welcome Prof. Septelia Inawati Wanandi. Professor, please.
Thank you very much for the nice introduction from the moderator. Good morning, everyone. Honorable guest professor, Professor Peter Lobie, welcome to our faculty. Thank you for sharing us your inspiring knowledge and research experience. Thank you very much. Esteemed professor in this faculty, Professor Hariyono Winarto, also a speaker today, Professor Wawai Muli, and other faculty member, researchers, students, and all audience offline in this room and also online. I would like to begin by expressing sincere gratitude to the Master Program in Biomedical Sciences for extending this invitation to me. Today I would like to share, I'm honored to share our journey on the application of CRISPR-Cas9 system as a transformative approach for the advancement of breast cancer therapy. I'm from the Department of Biochemistry and Molecular Biology, also from the Molecular Biology and Proteomic facility in this institution. So, slide please. So, we are going to talk about moving from fundamental laboratory-based research in vitro, in silico, and also in vivo to the goal, toward the goal of bedside precision oncology. So what we know about the traditional genetic engineering with the pioneer like Stanley Cohen and Herbert Boyer in 1973, slide, and by, slide please, 2007, slide please, slide. Who handled the slide? Sorry. Okay. And slide. By 2007, the creation of knockout mice had earned the Nobel Prize for physiology, proving that we could delete and or mutate the specific genes to understand the pathophysiology of disease. Slide. Both work were the traditional genetic engineering. Then came the era of genome editing. We define genome editing as the manipulation of genetic material of a living organism by deleting, inserting, or replacing the DNA sequence within the genome. Using the engineered restriction endonuclease or meganuclease to cut a specific target of DNA. This pathway of the genome editing, we can knock out or knock in the expression, the gene expression, with the aim of improving a crop, farm, animal, or correcting a genetic disorder. Compared to the traditional genetic engineering that practice the transgenesis, where the new gene can be inserted into a cell, but it was random. It means that it can land it randomly in a wrong spot, so the new gene can be inserted in a healthy gene and can cause the cell become cancerous. So in the genome editing, the target is specific using the guide RNA, which then direct the enzyme to the precise location within the genome. Slide. This is the meganucleus for genome editing compared to the earlier tools, single nuclease and talons. CRISPR-Cas9 system has superiority. So in using the RNA base compared to these two earlier tools using protein base, which require engineering complex protein to direct the nucleus. But in CRISPR-Cas system, we use RNA base that is more simpler and more efficient tools for the genome editing. Slide please. So in 2020, Jennifer Doudna and Emmanuel Charpentier had earned the Nobel Prize in Chemistry for CRISPR-Cas9. This is recognized as, at the moment, as the most efficient tools available. Slide. So this is the CRISPR-Cas9 system that need the single guide RNA to direct the Cas9 enzyme to the precise location and cut as a molecular scissor cut in this target. Slide. Okay, then after the Cas9 cut the target, then come the adaptive response of the cells, which is a DNA repair mechanism, and recognize the double-strand DNA break that already generated by the Cas9 enzyme. And this system can use the cell DNA repair mechanism, which is non-homologous end joining, to generate the knockout cells, knockout genes in the cells, or we can also direct to the knock-in system. Slide. So basically, the workflow of CRISPR-Cas9 genome editing involves several rigorous steps like determining the target DNA fragment in the genome, designing the sgRNA sequences, preparing the CRISPR-Cas9 complex, evaluation of cleavage activity of the complex, transfecting the plasmid, CRISPR-Cas9 plasmid into target cell, and finally detecting and confirming the indel mutation in the target cells. Slide. So designing sgRNA sequences, we can use bioinformatic tools. Slide. And slide, please. Yeah, and preparing the CRISPR-Cas9 complex via, we can choose expression plasmid or active protein. We can directly send it in direct active protein, mRNA, or viral particles. Slide. So next we have to evaluate the activity, the cleavage activity of this CRISPR-Cas9, and we can use, slide please, next, yeah, we can use this assay to compare the activity, the cleavage activity of the CRISPR-Cas9 system using EGFP protein. Slide. Then finally we have to confirming the indel mutation, insertion and deletion mutation using PCR or DNA sequencing. Slide. Now I would like to share about our project. This project are conducted in the molecular biology and proteomic core facility in this building, in this institution, IMERI FKUI, with the aim of targeting specific genes responsible for survival and stemness for precision oncology in breast cancer therapy. Slide. So two genes are ongoing now. The first is superoxide dismutase 2, SOD2, or we call also as manganese superoxide dismutase. This enzyme is actually antioxidant enzyme, normal antioxidant enzyme that is very essential for the antioxidant system in our body, and located in the mitochondrial matrix. Slide. Our previous study have found that these enzymes are highly expressed in human breast cancer stem cell, and this is linked to their survival and cancer stemness activity. Slide. Then we organize the gene and also the protein structure. We found there are 9 variants of human SOD2 protein, which commonly use the last half of exon 2 region. So this is the exon 2, and we found also the site, the target site for our editing. Right? Slide. Yeah, this the research flow begin with the determining the conserved region among 9 SOD variant in the last half of the exon 2, and then designing the sgRNA for knocking out SOD gene expression, and we evaluate the cleavage activity, which show higher signal fluorescence compared to the wild type, and then we confirming the activity, the expression, mRNA expression and protein expression, which is reduced, and also the activity of SOD2 are suppressed by this knockout system. Next. So we evaluate the point mutation and insertion mutation we found in the sequence, DNA sequence, the indel mutation in the sense strand as well as in the anti-sense strands, either deletion, insertion, substitution. Then, slide. Yeah, we predicted also the structure, 3D structure using Swiss model and also PyMol, and show that the substitution of asparagine 63 to threonine 63 and valine 64 to phenylalanine changed the conformation of the protein from helix, alpha 3 helix turn to strand. And also we found that the substitution of threonine 65 to asparagine 65 did not affect the protein conformation. Slide. Yeah, these are the substitution. Slide. As for the insertion mutation, we found that this affect the active site of the enzyme that will affect also the binding of the substrate to the enzyme. Slide. Yeah, this one. Slide. Slide. Yeah. So what is the impact of this knockout SOD2 in the triple negative cells? We use BT 549, and we using the protein array with 26 protein, we could suggest that this knockout SOD2 affect the cell stress response. We are using the panel for cell stress response protein, which show that some protein are upregulated and some are downregulated. Slide. Then we also analyze the reduction in cell proliferation using cell cycle phase, cell proliferation assay, and also the increase of population doubling time. Slide. We also demonstrate the impact of KO SOD2 in TNBC cells, which increase the apoptosis. Here, tunnel assay, we showed either in early apoptosis phase or late apoptosis, both are increased. And also the apoptosis protein, the pro-apoptosis caspase 3 and caspase 9 are increased. And also the survivin is the anti-apoptosis is suppressed. The expression was suppressed. Next slide. KO-SOD2 in TNBC cell also reduce the cell stemness by reducing CSC population, as shown here, from 50% to 14% in the knockout cells, and also the pluripotent cells such as OC4 and SOX2 and also NANOC. Slide. This project has been published in post-oral and poster presentation. Also have one registered patents has been registered, and 2 original articles has been published. Was conducted by Sekar, one of the laboratory research assistant, and also master students in master program in biomedical sciences. Slide. Now the second gene that we are going to discuss is survivin. Survivin is the gene for survival. So slide. This is the inhibitor of apoptosis protein or IAP family. And survivin is highly expressed in embryonic state and also cancer, but nearly absent its expression in normal adult cells. So in breast cancer cell, based on previous study, the highly aggressive breast cancer subtype, triple negative, express the most highest survivin expression compared to the other subtypes. Slide. So we select which site that we want to edit for survivin and found that exon 2 that are conserved in all these 6 alternative splice variants encoded by BRCA5 gene, the gene for survivin, and exon 2 comprise also the BIR domain. BIR domain is the active site of survivin. So it is important if we want to knock out the survivin, we targeted this exon 2. Slide. So in this project, we use dual sgRNA approach. Means that 2 sgRNA were designed to delete exon 2 of human survivin gene. We hope that we could cut in this site and this site so that the entire exon 2 can be deleted. Slide, please. And then we confirm the knockout of survivin in BT549 TNBC cells and shown that all this is the original protein has been deleted in the, this is the DNA, sorry, and the protein also has been deleted, cannot be expressed. And we confirm with the DNA sequence that have I forgot 1,100 base pair deletion. So the entire exon 2 has been deleted using the dual sgRNAs approach. This is one of the advantages using the Cas9 because we can CRISPR-Cas9 we can do it multiplexing, means that more than one gene can be mutated at once. So that we can target more genes at once. Slide. Then prediction of 3D structure. This is the original survivin, and this is the knockout one. That 40 amino acids compared to the 142 amino acid, leading to a complete loss of its function, including the BIR domain that I already mentioned. This is the active site of survivin. Slide. Yeah. So we also analyzed the impact of this KO survivin, shown that the KO survivin cells were arrested in G1 genol and G1 phase, with reducing cell proliferation. You see, and also the cell cycle showing that the genol and G1 increase. Slide. So other is the apoptosis, which KO survivin, actually survivin is anti-apoptosis. So if it is knocked out, so it will increase the apoptosis. We confirm in this assay, apoptosis assay, and showing that the early and late phase of apoptosis are increased in the knockout cells, and also protein responsible for the apoptosis. So, and next is the effect on cancer stemness. We saw that the cancer stemness were reduced. We showed here through the population of ALDH1 in the knockout cells, reduced compared to the wild type, and also tunnel assay and the mammosphere forming assay, showing that the stemness are reducing using this KO survivin. Slide. So an analysis also using PPI, protein-protein interaction on stemness protein, and we suggest that survivin regulates this stemness factor indirectly through a network involving P53 and CMYC. So this is the proposed mechanism that we found through this analysis. Slide. Then for the cellular stress response, we can analyze the genotoxic using this panel. We can analyze the genotoxic stress, hypoxic stress, and also oxidative stress, which are increased, and also increase the Ross level of superoxide, which is actually beneficial for the cell death, and to decrease the survival. Slide. So finally, we analyzes the metastasis protein, and we found that KO survivin in TNBC cells affected the metastasis protein, reducing the some protein like pimentin, CMAT, and HGF protein that is responsible for the metastasis. Slide. So after looking at the band system, in vitro system, we are moving to the living system. Using the CRISPR-Cas9, it is allowing us to moving to the in vivo validation and delivery and validation of knockout survivin in the MBA-induced rat models. We inject CRISPR-Cas9 plasmid in lipid nanoparticle complex into mammary tumor nodules. Before that, we designed the dual sgRNA, also for mammary gene, for rat genes. Slide. Then we found that we evaluate the cleavage activity in mammary tumor nodules using the EGFP protein, the signal, fluorescent signal, showing that the tumor nodule has been transfected with the CRISPR-Cas9 plasmid containing the sgRNA. Slide. So we show that dual sgRNA approach in CRISPR-Cas9 system could reduce the survivin expression in mammary tumor, even though it's not whole entire reduced, but partially reduced. Then we analyzed the effect on the slide. The effect on the body weight change and also relative tumor volume. The rats treated with dual sgRNA showed reduced tumor volume and healthy body weight gain, suggesting both the efficacy and potential safety of this targeted treatment. Slide. So this project has been published, performed by Rasda, one of our research assistants, now is already holding the PhD degree, and also Sylvia Tun from the alumni from the master program in biomedical sciences. So we got, as the output, we got 1 patent registered, 3 original article, 1 review article, 1 oral presentation, and 1 poster presentation, including awards for Rajak Awards in 2022, Young Scientist Award in 2024, and Falling Walls Lab from DAAD in 2025. Slide, I think the conclusion. So we have discussed, I have shown you the transition from traditional genetic engineering to genome editing is the transition from general modification to precision gene editing. CRISPR system is widely considered the simplest and the most efficient editing tool ever developed, and allow us to perform multiplexing, more than 1 gene targeted. In our survivin study, the use of Cas9 and dual sgRNA approach significantly increase specificity and ensure the gene is completely deleted. The ease of programming CRISPR system allow us to move rapidly into the in vivo rate models and potentially more further into clinical trials. We also have to consider the ethical consideration should distinguish between somatic therapy to cure patient, like what we do in our project, and the controversial germline editing. This will be the ethical consideration, and our goal remains to refine this system to make precision gene therapy a safe and effective reality for cancer patients. As acknowledgement, next slide, I would like to send special thanks to Universities of Tsukuba for tech transfer of this CRISPR-Cas9 genome editing in the beginning of our study, and also for Emery FKUI, especially the cluster molecular biology and proteomics facility and animal research facility. Slide. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Prof. Ina, for the very enriching presentation. This insight have given us a deeper understanding in molecular and translational research, of course. So let's proceed to continue to our third speaker. Kindly please the slide of the curriculum vitae. Yeah. So it's our honor to introduce our third speaker, Professor Dr. Hariyono Winarto, Specialist Obstetrics and Gynecology, Subspecialist Oncology, from Department of Obstetrics and Gynecology, Faculty of Medicine, Universitas Indonesia. Prof. Hariyono Winarto earned his doctoral degree in Biomedical Sciences from Faculty of Medicine, Universitas Indonesia, after completing his medical degree and specialist training in obstetrics and gynecology at the same institution. He has long been dedicated to academic and clinical service in gynecological oncology. He currently serves as the lecturer of Department of Obstetrics and Gynecology FMUI, staff of the Gynecologic Oncology Division, head of Gynecologic Oncology Division, and also vice Director of Institute Development at Emery FMUI. His research and publication have contributed significantly to the field of women's cancer presentation and management, including studies of HPV awareness and cervical cancer prevention in Indonesia, cervical cancer elimination strategies in Asia Pacific region, and molecular predictors of radiotherapy response in cervical cancers. We are very pleased to have him with us today. Please join me welcoming Professor Dr. Hariyono Winarto, Specialist Obstetrics and Gynecology, Specialist Oncology. Professor, time is yours for 20 minutes.
Yeah. First of all, thank you for the committee who invites me. It's an honor for me to talk here with very distinguished speakers like Professor Lobie and Professor Septelia Inawati Wanandi. Thank you, Professor Wawemuli, for arranging this meeting. Okay, yeah, thank you. I think I will talk about the reality in clinical situations, about the clinical situations and clinical epidemiology of ovarian cancer in Indonesia. Next slide, please. Okay, actually, globally, the ovarian cancers ranks 8th, but in Indonesia, for female, it is the 3rd. So, not the lung cancer. Okay, yes. Slide next, please. Okay, and the incidence, I don't know, perhaps this is also the increase is because of our government insurance is better now. So many patients comes to the hospitals, to the clinic. So it looks like that, it looks like that the incidence is rise. Okay, this is the absolute volume. What is the amount of the incidence? Actually, in 2021, we have about 13,250 ovarian cancer cases. To be honest, it could be more because not every Indonesian patients come to the hospital. So this is the amount of the patients registered in the hospital. And the highest rate is in West Papua. 11.2 per 100,000 people. Next slide, please. So this is the most of the patients who is diagnosed as ovarian cancer is between the age of 50 until 54. Although we have also very young age, and postmenopausal woman is the peak of the incidence of the ovarian cancers. And the menopausal age in Indonesia is around 49 until 42 age, but some also could reach near the 60. Okay. Next slide, please. So this is the clinical presentation, what we diagnose in the hospital. And to be honest, most of them, we don't know the states. Why? Because the refer come from peripheral area done by, not always by the OBGYN, but also by the general surgeon or the general OBGYN. And as Asian people, they don't want to have another surgery to be states. Actually, it was states by surgery. So they don't want, so we don't know the states. And also the follow-up here in Indonesia is a bit difficult to follow up because they move always. The patient always move. I mean, they don't have the accommodations in Jakarta, so they always move. So sometimes we could not easily follow up the patients. But most of them is stage 3C, sorry, it is mistype. So the most of the patients is serous carcinoma. I don't know whether in Shenzhen is a serous carcinoma also. Yeah, and this is still a problem because most of the case is relapse and resistance, and survival is very low. Although the other like clear carcinoma we have about 8.17%. I think this is a bit bigger the percentage than Western people for the clear cell carcinoma. And endometrioid carcinoma, we have also more now, about 6.20, and mucinous also we have more compared to compared to the Western populations. Next slide, please. So this is the survival. It is still very, very low. While this hospital is a government hospital, we have the government insurance for the chemotherapy, and it takes quite times for giving the chemotherapy also. And well, the refer also is already doing operated surgery, and they don't want to be operated again. So it also make the survival is even lower. It's about only 7.4%. This is for the serous carcinoma, of course. But for the clear cell carcinoma, it is better, a lot better. Yeah, it's 70%, and for the mucinous is also a lot better. And you know, the characteristic of our people, Indonesian people, they don't want to come to the hospital until the abdomen become big. So it or before there is signs, clinical signs. So if there is already clinical signs, it's already advanced stage. So because we are a referral hospital, so our survival rate is also very low. Next slide, please. So this is the clear cell. Okay, next slide, please. So this is the survival outcomes. Yeah. So I think the clear cell is better than the serous carcinoma. Yeah. Slide, please. Okay. Then also, I think we are a very good site for others. If you want to have advanced stage of ovarian cancer clinical trial, because now we could we still have a discussion to have with Etana for what do you say, the PD-1 clinical trial, because of the most of the patients is already advanced stage. Okay. Next slide, please. So this is the grade of the across tumor differentiation. This is in general. So of course, if it is more, then the survival is less and less. Yeah. Another slide. Next slide. Yeah. This is the one-year survival and median survival time based on degree of differentiation. I think. Yeah. Slide, please. The unknown means that it was very poor under microscope to from the pathologies, and we could not classify it again. Is there any pathologists here? No, yeah. Okay, so this is an iceberg phenomenon. it's not only in infections of that we have iceberg phenomenon, but so this is also the ovarian cancer. Actually, we have iceberg phenomenon. So this is who come to the doctors, and the other is they don't come or come in a late stage. Yeah. Next slide. Yeah, we have 240 until 250 million. We have only about almost 5,000 obstetrics and gynecologists. And of course, because Java Island is very populated, 55% is in Java Island, and we have only 121 gyne-oncologists in across the country. So we still push for the amount of the specialists in Indonesia, of course. Slide, please. Okay, I think it's the big picture of the ovarian cancers. So thank you very much. Yeah.
Okay, thank you very much, Prof. Hariyono Winarto. First of all, let me please kindly invite the three speakers to sit in front of the here. Can you please sit here? Have a seat. Okay. So actually, we have online and on-site participants, dear professor. So for the first round, for the question and answer question, for the first round, I would like to give chance to the online participants. Is that okay? Okay. So can you please? Is there any the committee? Is there any question from Zoom? Not yet, yeah. Oh, okay. So, okay. Because from the online, there is still no question. So I would like to give the first chance to Pak Ari. Yeah.
Okay, thank you, Dr. Ria. Okay, thank you for the nice lecture for three of you. Okay, first I would like to address my question to Professor Peter Lobie. Okay, so you explained that actually using the inhibitor of PET phosphorylation can be used to new therapeutic agent for the female ovarian cancer. Okay, I think the AKT pathway is a common pathway to other cell type also. So do you think that by giving the PET phosphorylation inhibitor can be applied to other cancer? So that is my first question. And then you explained that you using the PET serine 99. It's very specific location. Why is that? So what would happen if we just giving the inhibitor for the global phosphorylation for the PET phosphorylation, for example? So why very specific to PET serine 99? Okay, and then very interesting phenomenon that when you apply inhibitor to PI3 kinase and PET phosphorylation, and then PIM1 kinase step in. So what mechanism to stimulate PIM1 to step in? So that is very interesting. And then to Prof. Ina, so very interesting also for the CRISPR-Cas9. So,
Prof, maybe so we can still remember your questions.
so
we can
you answer it.
we can take one by one. So the first question was, could we use a BAD inhibitor in any other cancer type? Definitely. In fact, we have a number of different models which are not gynecologic, where we can use the BAD inhibitor. So basically, any cancer type where PI3 kinase pathway or MAP kinase pathway is overactive would be a very useful application of a BAD inhibitor. One we've looked at, but we haven't published yet, it's in Cell Reports Medicine under review at the moment, is in lung adenocarcinoma treated with EGF receptor tyrosine kinase inhibitors. So they also initially decrease bad phosphorylation, but then resistance develops, and in combination, a BAD inhibitor with an EGF receptor tyrosine kinase inhibitor, again, we're able to obtain a complete response in patient-derived xenograft models. Another good example would be in pancreatic adenocarcinoma, pancreatic ductal adenocarcinoma, where most of these cancers have a KRAS mutation, which hyperactivates the MAP kinase pathway. So we've shown that if we interrupt this hyperactivation in combination with a bad phosphorylation inhibitor, we're also able to actually produce regression in patient-derived models, which is also really quite unique for pancreatic ductal adenocarcinoma. It's very, very difficult to treat. And there's a number of other models we've looked at. Tamoxifen-resistant ER-positive breast cancer is one. There's a couple of others. So the second question from memory was why specifically bad serine 99? So bad actually will bind to 1433 and take on a survival function when both serine 75 and 99 are phosphorylated. If serine 75 phosphorylation is missing, it will go pro-cell death. If serine 99 is missing, it will also go pro-cell death. So we only need to inhibit one, phosphorylation. We do it in a non-kinase dependent manner because the kinase inhibitors will reduce phosphorylation generally, but eventually resistance will develop. And I think that's been the case with PI3 kinase inhibitors, with inhibitors of the MAP kinase pathway, for example, or with receptor tyrosine kinase inhibitors. So when this escape pathway happens, bad serine 75 and serine 99 phosphorylation are maintained. This happens, we think the primary reason is because of PIM1 kinase. What produces the upregulation of PIM1 kinase activity, we're not sure. We do know it is JAK kinase dependent, right? JAK kinase is a non-receptor tyrosine kinase that can also bind to receptor tyrosine kinases, quite independent of their ligand binding, and activate the rescue pathway. I always think of cancer cells like individuals in a human population. Everyone's always looking for ways to survive. And some people in a population come up with good ways to survive. So cells that choose PIM1, it appears to be a good way to survive. Exact mechanisms, we're not sure, but we know if we inhibit PIM1 kinase and BAD together, the response is very, very good. Thank you.
Thank you very much. I guess my question to Prof. Ina. So in your model, knockout SOD2 and also knockout for sulfivin, you mentioned about the effect also to the caspase-3, for example, and also for the so did you check all the cancer-related gene, for example, the oncogene and also maybe AKT pathway, so and also for example, P53 for example. So did you check it?
We are using protein array panels that is commercialized. So we don't use proteomic as a proteomic bioinformatic dose because it's very expensive. So we have we bought a kit that is that was a proteomic array or protein array. Includes proteins that are responsible for the function, for example, cell stress response, for stemness, for stemness already, cell apoptosis, such as like that. Then some of the protein, including in the this array, this kit, is also protein that you mentioned responsible for the pathway. For example, NRF2 and P53 and so on. So then our researcher also analyzed the protein-protein interaction between proteins analyzed in this panel, using these panels. Is it answer your question?
Yeah, sure. Okay.
So not specialized in those specific protein that we analyzed. We found in the array analysis.
Okay, thank you. One more question about this is just technical question. So you check whether the CRISPR-Cas9 cleavage activity is good or not using the fluorescent
EGFR.
Why is that? I mean, why didn't you just check by PCR, for example?
So because we don't want to lose time if we not confirm the cleavage activity. If we already transfected, and this is time consuming and also money consuming, so we have to check first whether the plasmid that we prepare, that we design, is really good plasmid which have cleavage activity. This is not common, I know. It's not common. This was introduced by our collaborator in Tsinghua University, and this is very, I think it's very good to confirming that your plasmid is already ready for the next transfection.
Okay. Thank you.
Okay.
Okay, thank you for the question and answer, Professor. So let's move to the online question. This is from Wahyu Dewi. The first question for Prof. Peter Lobie and Prof. Ina. The question is, in your opinion, what is the best option to treat cancer for now? Is it targeted drug or gene editing? Is it already available for treatment now? And additional question for Prof. Ina, what is the chance of relapse in therapy using gene editing? Maybe the first round, I would like to give the chance to Prof. Lobie.
So I think both ways have advantages and disadvantages, right? One of the advantages of a small molecule approach is that you can be reasonably targeted, but most small molecules usually bind to other proteins as well, maybe of lower affinity, but it can also alter the functions of other proteins, so-called off-target side effects. The good point about a small molecule approach is it may be available orally, so it's much more convenient for the patient to take if they're on an outpatient basis. And also the tumor penetration is far superior to any gene therapy. And so it has the capacity to actually interact with every cancer cell in the body, regardless of whether it's a primary tumor or a metastatic tumor or a micrometastatic deposit in different locations. Gene therapy may have some delivery problems, and if you're not getting 100% delivery to cancer cells, then maybe some of the cells will escape and the tumor could potentially recur. But it does have an advantage in that it could be very, very specific, right? I think we saw, even targeting the different isoforms of survivin. So if you could identify one or two targets that are really specifically involved and optimize the delivery, gene therapy also does hold some promise. One area of, you're the expert, but one area of gene therapy that does hold a lot of promise is adoptive immunotherapy, where you can generate immune cells in vivo using delivery of nucleic acid constructs or particles, which don't need to therefore be delivered to every cancer cell, but they simply need to be delivered to at least a certain portion of immune cells, which will then change their function and specifically attack the cancer cells. So that's my idea on this. I'll now pass over to Professor.
Thank you very much, Professor Lobie. Thank you very much, Mewahyu, Ibu Mewahyu. I think I agree, Professor Lobie, that every treatment have advantages better than the others. Yeah. In this case, because I present the genome editing or gene editing, I would like to put a highlight in genome editing or gene editing. In this, my project is 2 genes, SOD2 and survivin. The gene editing is specifically targeted. Because we use the Cas9, using the Cas9, and also especially the dual sgRNA, so the hurdles of targets can be removed because this is specifically targeted. Even though we have to, I agree that the delivery matter is something that we should pay more attention in, especially in vivo or in clinical trial later on, so that we should ensure that no off-target sites will be affected using this gene editing. Especially for SOD2, we are concerned with SOD2. Since this is the normal antioxidant gene, this what actually needed for the healthy cells, for the normal cells. So if we delete the SOD2, the concern is that this will affect also the healthy cells. So the delivery is very important in our project targeted using the intratumor delivery, using the plasmid and mixed with the lipid nanoparticle complex to deliver this plasmid to edit specifically in the precise location of SOD2 in the tumor. But we haven't done. We haven't done with SOD2. In the next future, in the near future, we will try in SOD2. But for survivin, since survivin is nearly absent in the normal cells, so it's more safe than because we can target directly to the tumors. And your second question about what is the relapse?
The chance of relapse.
The chance of relapse with gene editing therapy. As we know that cancer is multifactorial disease with multi genes involved in the carcinogenesis. So meaning that suppressing one gene, it should be calculated which genes are as important for the whole mechanism of carcinogenesis, and this should be individually or personalized, targeted, because one patient may have higher expression of survivin, but the other patient no. So before using the gene editing, precise gene editing, we have to analyze which genes we would like to target to be edited. So this is if I cannot say whether if we do the gene editing, we can inhibit the relapse of the cancer, because multifactorial, multi-gene involved in the carcinogenesis. So thank you very much.
Okay, thank you very much, Prof, for the answer. I think it's very clear. For the online participants, can you can ask directly, actually. You can raise your hand and talk directly to our speakers. Is there any? Okay, but if you want to ask directly, whether the online and on-site participants, you are so welcome. So let's proceed to the second question from online participant, from Erwin Budi. Yeah, for the first question, for Prof. Hariyono. Regarding the iceberg phenomenon, are we getting closer to effective early detection of ovarian cancer, or is risk-based surveillance still the main approach? Yeah, maybe one question first. Yeah.
Until now, there is no early detection system established for ovarian cancers, only for cervical cancers and for breast cancers, because as we know that ovarian cancers is very variative. I mean, genetically different from each other, even in one type, even in seros. In clinic, we could find, although it is the same histology, the same grade of differentiation, and it could be difference in response of the therapy. So that what we face. And can I comment about the targeted therapy? Okay. So we have also experience with the targeted therapy in clinical settings. For example, the bevacizumab. This is a very classic anti-angiogenesis. For some patients, we could give the bevacizumab for a very long time, and then at a point of time, it will not respond anymore, and then we try the others. For example, in other case, for example, is cervical cancer. Cervical cancers in clinical use, also we could use the bevacizumab, and then we use the pembrolizumab. The cost of the pembrolizumab is very high, but perhaps Peter Lobie know the answers. At a point of time, then it would not work anymore. It's not about the targeted therapy, but also every time we give the chemotherapy, for example, the first line, and then it come the response is not response, and then we give the second line, it's not response anymore, and then the other regimens seems to be not works also. So then yeah, that's the real problem in clinics, because we have because we are a national referral center. Then all of the difficult and advanced stage cases come to us. So that's still a problem for us. So what do you think?
Yeah. Is there any like a combination between the therapy? If the first line is not working, is it directly go to the second line?
Yeah, to be honest, it depends on the insurance also. So if the insurance could also for targeted therapy, then we could give, but not all of the patients have good faith.
So actually we have BPJS in Indonesia.
Yeah,
That is widely covered, but not all the regimen is covered by the BPJS. Yeah. Okay. I think it's so very clear and gave good insight for us. The second question from Erwin Budi is for Prof. Peter Lobie. The question is, do you anticipate resistance will eventually limit the efficacy of BAD or BCL2 inhibitors, similar to other targeted therapies? And do you have any comments for new emerging targeted therapies, such as for alpha receptor? Maybe Prof. Hariyono also can give comments here to this question. Okay, first Prof. Lobie.
Yes, I believe every drug used as a single agent has the capacity to develop resistance. And I think you saw that on some of the graphs that I showed in the talk, whereby use as a single agent, it showed efficacy, but it didn't end up in a complete response. So I believe not only designing new drugs to new targets is good, but the real answer lies in how you combine existing or new drugs together in a combination such that a much better outcome can be achieved for patients. And I think it's pretty clear why combination should be much more effective. Yep. And now,
is there any like new emerging targeted therapy for like alpha receptor for any kind of cancer type? Or maybe you can give one example for ovarian, or maybe
yeah. I mean, this is a first-in-class drug that we were working on already, so it's a new target. New targets in China are actually very difficult to develop because industry is very reluctant to take on the risk to develop new targets and new drugs, and they simply prefer to make me too or me better drugs. I think Professor Muli, on another day I also have another talk. I'll introduce another target, another new target that we are working on, also effective in a range of different female reproductive cancers, especially breast. and our lab has two other new targets which I won't talk about on this trip. So I think there's a lot of laboratories that are looking for new targets. It's not a simple matter of finding a new target, but it's a matter of finding a new target that is also extremely potent, right? So when you inhibit it, the therapeutic effect will be also much more potent. Everyone has their favorite new target, but actually objectively identifying new targets that could change the outcomes for cancer patients is really what is required. And I think everyone will recognize, drug companies are not very innovative in general. Someone comes up with an idea to develop drugs to PD-1 or PD-L1, and then 25, 30 other companies will do the same thing. When it's probably not really necessary and actually in the end not financially viable. It would be actually better if they took risks on different new targets.
Is there any comments from Prof. Hariyono, Prof. Ina? Okay. From the clinical side, maybe? Maybe you can suggest BPJS to focus on.
Yeah, I do agree with a single agent, although it is a chemo or I think it is not bad, but the multi-agent is better. You know that every cells in tumors, perhaps, as Professor Peter Lobie said, want to survive, and if you know about the clonal theory in the cancer cells populations, so they will try to modify themselves to run from the immune system, from the therapeutic targets. So if we only target in one side, for example, the very classic old one is the anti-angiogenesis, then of course it will at some point not respond anymore, because they already know how to hinder the anti-angiogenesis. Also the anti-PD-L1, anti-PD-1, at some points it doesn't work anymore, although it is very, very expensive. Yeah, very expensive. People can sell anything to buy these drugs, but it doesn't work anymore. Okay. Yeah.
Okay, thank you. So let's move to the on-site participants. Is there any other question? Maybe from the lecturers over there? Dr. Vivian, Bu Ari? Okay, let's give Dr. Rizky. Yeah.
Thank you very much. Thank you very much, Dr. Ria, for the introduction, and I'm really honored in this forum. My name is Muhammad Rizky Fadila. I'm from the Department of Pharmacology, and also I'm a doctoral student supervised by Prof. Molly, Prof. Peter Lobie. It's nice to meet you. I have a question, but actually Prof. Ina has told previously that one of the novel therapeutic methods like the CRISPR-Cas9, even though it's precise, but has some potentially of toxicity, right, Prof. Ina? And also Prof. Hariyono has told to us before that, like the major problem when we translate our findings might not also because of not the therapy is not potent, but because of the good faith of the patients, right, the regulatory and etc. My question is directed to Prof. Peter Lobie. Actually, Prof. Peter Lobie, I'm really interested with your previous research, and I also heard from you that you explore the kinase, the potential kinase therapy. I'm also a medical doctor. I want to ask one simple question. How is your experience in translating your findings? Yeah, you have found the best compound, and also maybe in your experience, this compound has a minimum side effect potentially. How is your experience collaborate with industry or maybe the clinical researcher, medical doctor in the maybe the collaborator of the Tsinghua University? That's my question. Thank you very much. I want to learn from you. Thank you.
Thank you. I think to collaborate with medical doctors is very easy, especially on an experimental basis. Collaborating with industry coming from academia is a whole different game. Yeah? A whole different game. A whole different skill set and a whole different set of requirements or expectations. And, you know, to be frank, it's very, very difficult. You need to be very, very persistent. Yeah? You try, and reject. It's worse than trying to get a paper published. Because money is involved, and the people who are putting the money on the table, their outcome is not papers or clinical benefit. Their outcome is money, right? If you look at it very coldly. So especially in China now, you've probably heard of it, there's this massive AI revolution impacting on almost everything we do. And Shenzhen, although it's a high-tech city, even the government has funneled a lot of the money into very specific areas. And the investors are also funneling their money into very specific areas. And they're areas that, they would like to consider it's relatively low risk, but high return and rapid return. Doing drug development projects is a little bit longer term, as you would be aware as a clinical doctor, highly expensive and highly risky. So in general, there is some reluctance. Given that a number of organizations, mostly government funded, have been set up now in China. One example is the Lingang lab in Shanghai, whereby basically they'll take basic discoveries and push it all the way through to phase 1. It's an enormous commitment from the Shanghai government. Their budget is just phenomenal to do this. So I see some of these sort of semi-government ventures being much more rewarding for an academic or a laboratory scientist to be able to translate through to industry. Industry in China wants to come in when the project is more mature, when they think they can get a lower risk and a better benefit. So it seems the government is sort of stepping in and covering the gap. And to develop this drug, we're in contact with some of these people to progress that way rather than going straight on a commercial path. Yeah.
Thank you, Prof. Yeah, I think it's a common thing that happened. Yeah.
Everywhere.
I think that it's. Thank you. Oh, Prof. Prof. Hariyono.
Oh, no, I think it's okay. I would like to ask Prof. Lobie, is it okay? Okay. Following the question of Dr. Ari, Bapak Ari, that how we determine the site of phosphorylation that we target our small molecule. So I just, because we have done before, that we are using bioinformatic analysis to simulate the small molecule interaction with our protein. Then with this molecular docking, we could demonstrate that this small molecule inhibitor can directly interact with certain site of the protein, certain amino acid of the protein that will be phosphorylated to be activated, so that we can assume that this phosphorylation will be inhibited by this interaction of small molecule to the protein. So I would like to ask whether this approach is another approach that you use, or is this approach can be used to analyze?
So informatic prediction is a tool that can be used, but it's a prediction in the end. Often it's not accurate, even with the advanced AI tools that we're using today, but it can give an indication. So we really go back and do a lot of the molecular biology to prove the specificity. For example, a cell actually doesn't need bad to survive because it has both a pro-survival and a pro-death function. If we knock out bad, the cell survives quite fine. So it's the ratio of phosphorylated bad to non-phosphorylated bad that determines the outcome. So then we can knock in wild type bad, and we can restore the sensitivity to the drug, because then you both have phosphorylated, non-phosphorylated. Or we can knock in bad with a serine 99 mutation. And we see that the drug doesn't work. Okay, yeah? You know, and for our more potent drug, you know, it actually binds to more molecules than bad. You know, even if you can predict it might bind in the vicinity of serine 99, the ultimate proof is experimental, yeah? And so we spend a lot of time and effort, going back and doing all these studies to show that this one specific amino acid is responsible.
Maybe from online first, yeah. Sorry. Okay, actually there is still one question from online participants. This is from Anna. The question for Prof. Peter: how does Tsinghua University integrate multi-omics approaches, genomic, proteomic, and metabolomics, to achieve system cells understanding in modern microbiology? How is Tsinghua preparing for future pandemics? I think it's a broad question. I think I opened it for the three speakers. Okay, first Prof. Lobie.
Yeah. Okay, so I'm not a microbiologist. I'm not a virologist. But from a scientific point of view, Tsinghua has set up a number of very sophisticated, well-funded omics laboratories. I think some people from University of Indonesia are already guests in one of those laboratories with Professor Zhang Linqi in Beijing, which does a lot of the vaccine and antibody therapeutic work. So once you've got a platform set up, whether it's an AI platform to predict epitopes or active sites, or a vaccine platform to actually generate new therapeutics, it becomes just a process, an iterative process, so to plug in a different type of virus if there's going to be a different pandemic coming through. So for a while there was a monkeypox scare, and they also started plugging in the monkeypox into their platform system. Yeah, and I think the good point about Tsinghua University, as you know, it's the #1 university in China. Depending on which rankings, you know, somewhere top 10 or top 20 in different subject areas globally. And it's also, you know, it's a very connected university with the government. As people get older, they also die of different things, right? And at different ages becomes more important. You're absolutely correct. Mutations do increase with age. Cancer is a disease of age. And often human cancers take 10 or 15 years to develop once they're formed. Why it occurs perimenopausal, I guess you could imply there's hormone changes. Prof, correct me if I'm wrong, but I believe polycystic ovarian syndrome patients also have an increased incidence of ovarian cancer, and they also show androgenization, an increase in male hormones. During and after menopause, females also show a relative increased ratio of male to female hormones when their ovaries also stop producing estrogen. So it could be something in the transition phase, the rebalancing of hormones that push ovarian cancer to the forefront at that age before it stabilizes again. Yeah? We're all on a one-way path. If you look at age-specific mortality, I think 55 to 60 for, I'll quote Australian figures, for Australian males, you have a 1% annual mortality. Once you get above 60, it's 2%. When you get above 90, it's 25%. So you will be constantly, the population will be constantly dying of different factors.
Thank you.
Thank you, Prof. Lobie and Prof. Hariyono. I think I agree with you that the hormonal imbalance in this period, age period, 50 to 54, just right after the menopause, and combined with the mutation, start the mutation. So both can increase the incidence of the ovarian cancer. After 55, for example, the transition of this hormonal imbalance is not dramatically anymore, not drastic anymore. So that's why it's maybe in this period is higher. Thank you.
Thank you, Pravina.
Okay, I think this is the last question. So for the distinguished speakers and our participants, ladies and gentlemen, as we come to the end of today's webinar, I would like to express my sincere gratitude to our esteemed speakers, Prof. Peter Lobie, Prof. Septelia, Inawati Wanandi, Prof. Hariyono Winarto. Thank you for your sharing your expertise, your experience, and your valuable insights with all of us today. So I would like also extend my appreciation to all participants, whether they are online or on-site. Okay, so let's give applause for all the speakers and all the participants. Okay, thank you.
All right, distinguished speakers, respected lecturers, and dear participants, both on-site and online, we come to the end of this session. I would like to extend our deepest appreciation to all our esteemed speakers for sharing their valuable knowledge, insights, and experiences. Your contribution have truly enriched our understanding and made this session both meaningful and inspiring. I would also like to express our sincere thanks to all participants for your active engagement, thoughtful attention, and enthusiasm throughout the session. We hope that the knowledge and perspective gained today will be beneficial for your academic and professional journeys. Thank you once again for your participation. My name is Shinta. I'm wishing everyone a pleasant day ahead. See you in the next event. Goodbye. I would like the committee to arrange for a photo session, so